Our next esteemed speaker, Dr. Paul A. Jensen, is trained as an engineer and microbiologist, and in 2016, he joined the bioengineering faculty at the University of Illinois to mentor students in systems microbiology, artificial intelligence, and automated science. His lab works to solve biological problems that are too large for either experiments or computation alone. They are able to do this through automation, artificial intelligence, and functional genomics, and his lab's deep phenotyping system has completed over 730,000 automated experiments since January 2020. With this automation, though, accuracy is extremely important, as he shares today how his lab uses visual cues to effectively communicate protocols and reduce errors. So without further ado, Dr. Jensen.
All right, thank you very much. That was a very kind introduction. I want to talk today about not so much the science that we do in the lab, but how we're using visualizations, including a lot of graphics we've made with BioRender to reduce errors that we have in the lab and in the classroom. So I'll admit I was a little intimidated talking at a conference about visualization. I was thinking my slides have got to be pretty good. I'm not the greatest in making slides, so instead I thought I'll make my slides pretty simple. As you'll see, because my first slide, I'm just going to talk about trash cans. So this is an image of a trash can that you've probably seen something like it. The trash goes in the circle right in the middle, and then the trays go up on top. And you might have wondered, like me, when you're trying to jam all your stuff at the end of your meal at a fast food restaurant, why do they make the hole so small? Like, couldn't you make it a lot bigger? It'd be much more efficient. You could put the food in faster. And there's a reason for that. The hole is small enough that you can't put the trays in. The trays have to go on top because they don't fit inside the hole. So there's some sort of method to the way we design trash cans, and it's not always a physical restriction. So this is another trash can. I spent a lot of time browsing commercial trash cans to prepare for this talk. So trash goes in one receptacle. There's a cue that the paper here goes in the long flat slot, because paper, newspapers, things you recycle usually look kind of like that, and only round things like bottles and cans can fit in the bottom. Now, there's nothing preventing you. You can throw the bottles in the wrong one, or you can roll up the paper and put it in. It's simply a visual cue that tells you what you should be doing to try and minimize the error. So this is actually a process. It's something we see a lot in quality engineering, and this idea of visual or physical cues to reduce errors has a name, and it's called poka-yoke. It was developed in Japan in the automotive industry to reduce errors, and we see it all over the place. So here's just a couple examples of poka-yoke, or design or cues to reduce errors. One is an electrical outlet. Obviously, you can only plug a three-prong outlet in one way. The ground has to be on the bottom in the United States. But also, you notice one of the slots is a little bit larger. So a polarized appliance that requires the electricity to come in one way, also has a larger left plug, so you know even if there's not a ground plug, it goes in on one side. This is an ATM keypad. So after you punch in your number, you hit enter, and the cancel, clear, and enter are color-coded like a traffic light. So most people will think to go ahead that I would press the green button. It's just a visual cue. Nothing's preventing you from pressing the wrong one, but it kind of helps. When I get in my car, I have to press the brake pedal before I start the engine. This is to make sure, for some reason, if something failed and the car was engaged, that I'm going to be in control of the vehicle. I'll have the brake depressed when the car turns on. So a safety feature by poka-yoke. And then finally, this is a favorite one. Whenever I go into Sam's Club or a store that requires a membership, they ask to see your membership card. And this always bewildered me, because they're not checking if I'm a member. I just need to show a card. It doesn't say if it's current or if I paid my membership for this year. So I finally asked them, why do you ask to see my card if you're not going to check that it's valid? And they said, well, a lot of people forget their card and don't realize it until the checkout when you need it. And at that point, it's a very difficult error to correct, because you're in line and you have your groceries and you have to go to customer service. So they just want to remind people to have their card and go back to their car and get it if they forgot it. It's not actually a security check at all. So lots of instances of this idea of poka-yoke. We have this in science. So I was thinking of examples of where we have design or visual cues to reduce errors. The classic one is gel electrophoresis. So making sure your gel goes in the box the correct way so the DNA doesn't run backwards off the gel. When I was taught to do electrophoresis, they said, remember, run to red. The DNA is always going to run down towards that red electrode. Now, still, there's nothing preventing you from putting the gel in backwards. A poka-yoke technique called pointing and calling would actually say we can do better than just having this mnemonic, this run to red. If people in your lab are having trouble remembering which way does it go, not only telling them run to red, but saying if you point to the red electrode and you say out loud, run to red, that's been proven in many studies to reduce errors. Right? Again, the technique is called pointing and calling. You see this is a picture of a train conductor in Japan. They do this. They point to the signs and say out loud, significantly reduces the number of errors. In the New York City subway, they have to point before opening the doors at a particular sign. They don't say it out loud, which isn't as good. The pointing still helps, but you can try that in your lab to say, point and say it out loud. Now, even if you have the gel in the right way, you still have to make sure you put the correct electrode, one on the cathode and one on the anode. So the electrophoresis equipment is color coded, that you put the black in the negative and the red in the positive. Not on this model, this is the one in our lab, but I think there are others where you can't actually turn them the right way. They're different shapes, so you can only plug the anode and the cathode into the correct spot. Here's my favorite example, and this will be great for whenever you're at a trivia contest and the category comes up for multi-well plate design. There's a lot of things that go into the design of plates that are cues to help out the experimenter. So on the side of a multi-well plate, this is the picture from BioRender, there are these chamfers. So if you notice up in this corner, I don't know if you can see my mouse here, there's a slight part of the corner that's cut out here. Now I checked on the plates in our lab, there's only one chamfer cutout in all of our different multi-well plates, but here the image shows that there's two of them, and this is to make sure when you take the lid off the plate that you can only put it on in one orientation. You can't accidentally turn the lid and put it on backwards because the chamfers line up only in one particular configuration. So I was curious if this is a standardized way to keep from making these errors. Why does the image have two and our plates only have one? So I looked up. This is an actual image from the ANSI SLAS standard. So every dimension on a multi-well plate is standardized. This is why the well spacing matches your multi-channel pipette, matches your PCR strip tubes. It's all standardized. The only thing that's not standardized, interesting, is this chamfer. So if it says up there, it says C note C, and it says the quantity and the location of the chamfers is optional. So if your friends ask, is every part of a multi-well plate standardized? Almost everything. They can have 0 to 4 chamfered corners. That's entirely up to the manufacturer, and that's the only thing that's left up to chance on a particular plate. Okay? But this is only for the humans. Robots don't have this problem. When they take the lid off, they don't turn the lid and have to look at the plate and put it on. It's just to reduce errors so you can cross-contaminate a sample when you relid the plate. I checked on our equipment. You could probably use this as a cue to make sure that the plate goes in the right orientation. So I looked at some of our plate readers and robots. Do they use this chamfer thing? This is a Biotech multi-flow. It has a bit of a chamfer on the upper left-hand corner, but this is just the part that holds the plate in. So I don't think that's actually a visual cue. I think that's just the way that arm comes in to secure the plate. The only piece of equipment in our lab that I found that has the chamfer, this is an Applied Biosystems qPCR machine. And it has a chamfer, you can see, but it's in the wrong corner. It's in the upper right, and all of our plates have the chamfer on the upper left. So I'm curious if other manufacturers can use this to help make sure that humans put the plates in correctly in their devices. Okay, so let's talk now about poka-yoke in our protocols. So this is examples, these are physical things that I've talked about for reducing errors. What about the process errors? How can visual communication help out humans as they go through and do science? So I'd like to convince you of 3 reasons why we should use this design or visual cues in our protocols. 1, it reduces errors. And this has been shown in numerous studies. This is an entire branch of quality engineering. It's a real thing and it helps. Secondly, there's a process to go about it. So I'll step you through how you should start to incorporate visuals to get the most bang for your buck. And third, I'm going to tell you about some of the work in our lab that the same process, these molecules work even at large scale. So this is a protocol used for a teaching lab. This is developed by some students, and Karen Jensen is a faculty in bioengineering here. And I want to convince you that just adding visuals to your protocol puts in a lot of hidden things that you wouldn't think about normally. It helps understand the flow, as we've seen other speakers talk about. You can communicate the overall picture, but I'm going to argue that there's smaller things. For example, in this protocol, in step one, they pick a colony off a plate and they put it into a 1.5 mil Eppendorf tube. Right? And after that, they add some different reagents and then transfer it into a strip tube, small PCR tubes. And there's an arrow from panel 2 to panel 3 that says those tubes are the ones that go in the qPCR machine. And if you've taught an introductory undergraduate lab, there will be students that try and jam the Eppendorf tube into the qPCR machine, which doesn't fit. So on a second level, besides understanding the flow of the experiment, this is a cue of which tube actually goes in the machine. And if you're holding the Eppendorf tube and walking to the qPCR machine, there's a cue in here that you're doing it wrong. It's actually the other tubes that need to go in, and you've missed a step inside the protocol. And so that's a second level beyond understanding the experiment, is the process, the steps that you need to go through to carry it out. Here's another example. This is again taken from the lab manual for that same course as our cell and tissue engineering course here at Illinois. I've shown the same information in two ways. So on the top, I've given you a table that says how you lay out an experiment. This has a DMSO control or an inhibitor that's put onto cells. And the students are setting up 6 different samples here. Again, exact same information, and tells exactly what well you should put it in, just as a table, or this is a figure I made in BioRender that goes on the bottom. Now, there's hidden information besides looking nicer in the bottom, because you get 2 other pieces of data. One is you should be using a 6-well plate, right? Because you could set up the top experiment in the corner of a 96-well plate, but this is a cue to the students that if you don't have a plate that only has 6 wells, you're doing something wrong. And second, again, the chamfers on the plate tell you which way the overall plate should go. So when you take the plate and you put it in the plate reader, you know you've kept the orientation correct, and your data are not going to be rotated or transposed some way because you set up the table correctly, but in the plate incorrectly. That's for a simple 6-well experiment. Imagine when you get up to larger experiments. This is another visual from a different lab where they're doing bacterial transformations with dilutions in both ways. They do 2-fold dilutions along the columns and 10-fold dilutions along the rows, and then they're supposed to plate that bottom set of 12 wells that are outlined in the rectangle. And you can imagine trying to communicate that process in words, right? Having the visual, having the whole 96-well plate with the correct orientation greatly reduces the errors. Now, you don't have to go in and come up with beautiful graphics for every process, like I've shown some of these really nice examples from the 202 lab. Even adding a couple images to your protocols can help. And I like to argue that a single image can improve your protocol in a couple ways. So this is a snapshot of a protocol from our lab for making glycerol stocks of bacteria. And what I want to argue is, let's say at step number 2, which is done in a centrifuge, if you just put a picture of a centrifuge in the margin next to that, there's a huge benefit from adding this one image. Number one, when you first pick up the protocol, at a glance, you can tell what equipment that you need. You don't have to read a separate equipment section or go through the individual steps. You can see all of the major equipment out there. And I'm very grateful that BioRender has tons of lab equipment. All of the boring things in the lab, gel boxes, centrifuges, they're in there as well, as long as all the interesting biology visuals. So you can find a centrifuge that looks just like the one in your lab. So you know what you need, you know what step you're on. So when you put the thing in the centrifuge and then you're going to go read ahead in the protocol, you don't have to scan to find what number am I on and maybe end up at the wrong step. You look for the picture that matches the thing in front of you and you pick up right there. Third, which centrifuge? We have 2 different kinds of centrifuge in the lab. Well, it's the one that matches the picture. So people who are just starting out in the lab don't have to say which centrifuge or stop and ask. They know that it's going to be this one, reducing process errors there. So just one image, just put in the corner, is a sign, is a cue to the person, and can eliminate 3 different types of errors in the lab. Okay, so how would I suggest going about improving the visual communication in the lab? So here are 3 steps, and these are from a process improvement methodology that we use in quality engineering. Step 1 is probably the most important and has nothing to do with the visualization. If you're going to use visual communication to try and root out errors, you need to create what's called a culture of psychological safety. Right? You need to convince everyone working in your lab that it's okay to make mistakes. Right? Because people will hide errors if they're afraid of being punished. If they think in a classroom environment, in your lab environment, if I mess up, my grade's going to go down, my advisor's going to be upset, they're not going to report the error. And errors that are not reported can't get fixed. So you have to tell people, we're trying to improve our process. You need to be very open and honest with every little thing that goes wrong, right? To help us improve, not so that we can punish you or say, you know, there's something wrong with you, because everyone's making mistakes. Teams that have good psychological safety, teams who don't feel like they're going to be punished, make fewer errors. This has been reproduced many times by different groups. And it's kind of counterintuitive. A lot of people push back on this, say, if I'm accepting of errors, if my people working in my group feel like it's okay to make errors, they're going to get complacent, they're going to make more errors, and the opposite is true. These groups end up making fewer errors because they fix things, they address them and bring them to the fore. Right? So that's step 1. You got to make sure your team is ready to talk about safety. Step 2 is figure out where you should be spending your time. And the easiest way to do this is by a check sheet. So on your protocol, say whenever you make a mistake, even if you catch it, even if you mixed up 2 tubes and you caught it and you put them back in the right order, whenever there's a small thing, just put a tally in the margin next to that step. Right? You can hang a sheet up in the lab and ask where with common mistakes, and people can put a check mark on it. That's all you need to do. Don't think about why you made it. Don't think about solutions. Just put a tally mark. If you're in a class, ask students to do this. Ask the TA that's walking around with the protocol to keep tally marks. Just count them up and record. That's all you need to do. Don't worry about fixing anything now. Then when you're ready for a quality campaign, when you're ready to address and improve your processes, you collect that data and you make what's called a Pareto chart. It's simply a histogram of how many times the different errors are made. And what you'll see is something that follows the Pareto principle. There are a vital few errors we keep on making all the time. And then there's a trivial many. There's a whole bunch of other errors that we see rarely, but there's a lot of them. And the key is to focus on the vital few. Because when you're trying to convince your boss that you should spend time improving your process, you want to say, look how many times we're making these errors, it's going to have a big effect. If you as a supervisor are trying to convince your employees to spend time, stop what they're doing, and focus on changing their process, you also need to say there's a couple things that we can do that are going to have a big impact. So focus on those few. That's our 3-step process. Now, just to finish up, I want to tell you that these same processes work at scale. So as was mentioned in the introduction, we do a lot of automated experiments. It's run off our deep phenotyping system, which does growth assays to reverse engineer bacteria. It goes on this flow chart where experiments are requested, a computer system translates them into instructions for robots, and they carry out the experiment, and then the data comes back in the end. Okay. Now, all of the system is monitored by 2 technicians who log in every day and see this screen. This guides them through everything they want to do for the experiments. Now, the technicians do not plan any of the experiments. Those are requested by other people. So they really don't know what's going on inside the experiments. All they do is follow these instructions. It has to lay out every step that they do. So it says, for example, this is one of our robots called a Mantis. Load these reagents. It has a unique identifiers so they can double check that they got the right reagent. It has the volumes that are listed right there. And so that's all laid out, all the stocks they need to put in. It says which plates they're supposed to put in, again, with unique identifiers. And then it tells them step by step, this is what you should be doing, with small visual cues saying this should be the temperature, this is the environment, so they can double check when they're moving these plates around the lab. Now, when we started off, we had just 1 or 2 robots. We did about 300 experiments a day. We're now up to 11 different robots, 8 different types of robots that you see up here, and we have multiples of some. We do about 10,000 experiments a day, again handled with a team of 2 technicians and 1 AI engineer that oversees the software side. So if you have all these experiments, where are you getting them and how are they managed? Well, our 10,000 experiments today are actually not designed by a human. They're entirely designed by an artificial intelligence agent that's using the experiments and then learning from them and planning the next day's experiments. So this leads to a big problem from a quality control perspective. Is normally a human who planned the experiment can look at the plate and kind of see if something overall went wrong. But in our case, there isn't any human who really knows what's going on in these experiments. And all the master planning is done by an AI agent in a computer somewhere. So how can our humans QC results that we don't have? Our solution to this is actually randomization. So we don't put our experiments in the plates in any order. Right? We use all 384 well plates and everything is randomized. Right? So all the experiments that are requested by different agents are mixed up and sorted out into plates. And this means our humans aren't looking for a particular pattern, they're looking for any pattern at all. Because if a pattern shows up, that's a problem. There shouldn't be any variation. We shouldn't have large clusters of things that don't grow or things that grow really well because everything is randomized. The only exception is that every plate has fixed controls. So you can see there's some blue spots in these plates. They're arrayed in a particular way as both positive and negative controls. And then on the right, all the data are plotted, and the orange points, the positive controls, should be high, and the green points should be down. They don't grow on the bottom, so you can get one check that way. But other than that, a spot check on the plate can tell you if there's a pattern, there's a problem, because we get around randomization. So that's visual cues, again, kind of the opposite, completely unplanned, randomizing your experiments can give you feedback when you're talking about experiments at scale. So to conclude, I hope I've convinced you of a couple of things around POCAYO. It's, again, a quality improvement methodology. It absolutely reduces errors. This is an entire academic field of study. Everything from ergonomics to human factors researchers have found that time and time again, these small, simple visual cues can reduce errors. It is a process, though. You need to go about it in a particular way, creating safety, finding where your errors are, and then attacking those areas to try and reduce particular errors. And this can work at any scale, from a single scientist working by themselves all the way up to doing tens of thousands of experiments with automated systems like we have in our lab. So with that, I want to thank all the people who helped with these visualizations. On the top, we have people who worked on the classroom side who developed these protocols and teach the lab courses and shared those with me so I could share them with you today. And then the bottom, this is our deep phenotyping team who managed the AI and the automation system and then developed our at-scale quality control protocols. So with that, thank you very much for the invitation to talk, and happy to take any questions.
Great. Thank you very much, Dr. Jensen. Really interesting. I had not truly considered the use case for visualization and communicating process and protocols. So thank you for sharing that. I think it was helpful for the team here. And I really like the concept of psychological safety. It's something that I've noticed has reduced errors in my line of work as well. So commend you for sharing that with the audience. We'll put some questions up here at the bottom, as you can see. I'm happy to read them for you, or if you'd like to address them directly yourself, you let me know.
Are there any POCAYOs that haven't been created that you think need to be created? Absolutely. One, the gel box thing, we haven't really solved that, getting that in the right way. I think there's a lot of good work that could be done with centrifuges, making sure that they're loaded correctly and that they're balanced. After the fact, a lot of centrifuges now won't spin if they're imbalanced, but stopping that before it happens, I think that would be those would be two areas in the physical lab that I would say we need some innovation in that space. Thank you so much, and I'm looking for the rest of the conference.
Thanks very, very much. Once again, Dr. Jensen, that was fascinating, really interesting stuff.